So this lecture will be covering Chapter 10 out of your textbook, which will be over muscle tissue. Now, muscle tissue is the last basic tissue type we're going to cover in this course. Remember, we've had epithelial tissue, connective, nervous, and now we're finally into muscle. Now, what sets muscle cells apart from other cells is the fact that they have contractility, which means that they can contract easily, and that's what they're specialized for. Now, these cells are going to come from the mesoderm layer of the embryonic development, and they're going to be using the actin myofilaments to undergo their contractions. Now, there's three types of muscle cells or muscle tissue that we need to know for this chapter. There is first the skeletal muscle, which is probably the one that everybody's most familiar with. Then we have cardiac muscle, and then smooth muscle. Cardiac muscle is characterized by having very long bundles of cells which have multi-nucleuses. So it kind of looks like there's multiple nucleuses in each cell. And they're also going to have what we call cross striations. Now, this is going to be the easiest way to identify skeletal muscle from smooth muscle. And these guys are going to do forceful and quick contractions, and these are the ones that you are able to control voluntarily. Now, cardiac muscle is a little bit different. It still has the cross striations that you find in skeletal muscle. However, these guys are going to be branched cells, and they're going to be bound together by intercalated discs, which we'll be able to see in the images. And these guys are involuntary contractions, and they're going to be vigorous and rhythmic. So you're going to be able to have almost a rhythm to them. Think of your heartbeat. And the last kind is your smooth muscle. These are going to be easy to identify compared to the other two because they don't have those nice little striations we see in the other two. And these guys are very slow, involuntary contractions, and they're going to be mainly in your GI tract. So here are the three types of muscle tissue. You have your skeletal muscle, and notice the striations going vertical here. That is going to be your striations, and notice that there's multiple nuclei in each one. Your cardiac tissue is going to have one nuclei. It's going to be branched, and then you're going to have these nice intercalated discs. And then finally we have our smooth muscle. Notice that there's no striations whatsoever, and they just kind of look like little almost mesenchyme cells. Now when we look at the actual images that you see under the microscope, you can see that you can still see the striations. The nuclei are definitely all in one cell, and same with your cardiac muscle. You can still see those intercalated discs, and the striations are pretty clear. And then finally you have your smooth muscle, no striations whatsoever. So the way contraction works in muscle cells is by sliding myosin filaments over actin filaments. Now myosin tends to be thicker than actin, so when we talk about it, whenever we show an image of it, the thicker the fiber, that's going to be your myosin fibers. The thinner ones are going to be your actin. Now, remember I told you this in Chapter 9, they start renaming some things that you already know the name of to different names for different tissues. So, in the case of muscle, we have cytoplasm, but we're going to call it sarcoplasm. The smooth ER is the sarcoplasmic reticulum, and then finally the cell membrane is the sarcolemma. So don't get confused whenever we see these on the exam, and they're just the same structure, just different names. So the first one we're going to go through is skeletal muscle. This is pretty much the gold standard for your muscle tissue. Now, the muscle fibers are very long, like we said earlier, cylindrical, multiple nuclei found in them. They tend to come from mesenchymal derived cells, and these guys are going to be called myoblasts. Now, remember, whenever we break up a word, if we see the word myo, that's going to mean muscle. And then blasts, remember, are the cells that are going to be the cells that they're going to derive from. So these are your precursor cells. All right. Now, As they're developing, they're going to fuse together to form myotubes, which eventually form your striated muscle fibers. Now, when we're looking at it under the microscope, you'll find your nuclei, they're going to be more elongated. They're not going to have that nice round characterization we see in other cells, and they're going to be peripheral under the sarcolemma. So right underneath the plasma membrane, basically, you'll find them and they'll be almost squished. Now, whenever we're looking at, or whenever we're talking about satellite cells within muscle fibers, these are kind of your stem cells. These are progenitor cells that will turn into muscle fibers if needed. So here's the general breakdown of how skeletal muscle is formed. You start with your myoblasts. We start fusing together, and we form our myotubes, and then eventually we form our muscle fiber with the multiple nuclei, and then we'll keep our satellite cell, which is our progenitor cell, in case we ever need to derive more. So we're going to go into how our skeletal muscle is organized within our body. So just like with every other section we've gone through, we've discussed the connective tissue that surrounds it and the names for them. Same here. Skeletal muscle isn't just muscle and that's it. There's going to be layers of connective tissue around them that are going to be important for protection and for structure and keeping in place. So the first one is the most outer, coming from epimysium. So the epimysium, that's going to be the external layer. It's going to be mostly dense connective tissue. And whenever we look at it, it's going to have septa that go inward. So what that means is it's going to almost form what appears to be a funnel-like structure. And this is where your blood vessels, nerve fibers, and your lymphatics are going to go into the muscle tissue. Then underneath the epi layer, we have our perimysium. This is going to be pretty much thin. It's just going to be around a bundle of muscle fibers, and we call this the fascicle. Now, the bundle of muscle fibers is going to have nerve, blood vessels, and lymphatics, and that's going to penetrate this perimysium into each fascicle to provide the blood flow, lymphatics, and any innervation from nervous system. The last layer is the endomysium. This is the most inner layer. It's going to be even thinner, very thin, very delicate, and it's going to be composed primarily of reticular fibers. There'll also be a few fibroblasts scattered around, which will be responsible for laying down the connective tissue of the reticular fibers. Now, this is going to surround the external lamina of each muscle fiber, and capillaries will be there to provide a nice rich environment of oxygen. So when we look at our image of a cross section of a muscle, you have your individual muscle fiber. Around that, you're going to have the endomysium. A bunch of these will come together to form a fascicle, and around that will be your perimysium. A bunch of fascicles will come together to form your final skeletal muscle, and around that will be our dense connective tissue forming the epimysium. So it's almost like each layer is being broken down and there's connective tissue around it. And here are those septa we were talking about with the epimysium, where we can have our blood vessels and lymphatics and nerves being able to penetrate through. So when we look at it under the light microscopy, we have our endomysium. So we have this is a cross section, so we're not going to see the nice striations we see when we look at it longitudinally. So the cross section shows you our endomysium circling one single muscle fiber. Then the fascicle will be surrounded by the perimysium, about like that. And then our epimysium is going to be at the end. Same here. We have our cross section again, and we're going to see our epimysium right about here. And notice that's going to be lined up. This stain is specially for reticular fibers. So this is going to light up those reticular fibers so that you can actually see that the endomysium is going to have primarily reticular fibers present rather than any other kind of connective tissue or connective fibers. All right, so collagen will be found in our connective tissue as well, and it's going to serve mainly to transmit mechanical forces that the skeletal muscle is causing whenever it constricts. So when we look at a skeletal muscle, you'll notice that they taper at the end. So if you're looking at a leg muscle, and pardon my drawing, but it almost looks like this, where it tapers at the ends of the muscle and it's thicker in the center. Now, the epimysium is going to be continuous all the way through and eventually connect with our tendons. And we call these myotendinous junctions. Myo meaning muscle, tendinous meaning tendons. And then junctions mean that's where they're going to come together. So, here's our muscle, and then our tendon. Our muscle is going to be identified with the M. Our tendon is identified with the T. And you notice that the epimysium is going to be all the way through and continuous for the whole stretch of the muscle. Alright, so this is probably one of the more important parts of the muscle fibers, and I can guarantee you this will be on the exam. But we're going to go into how each muscle fiber is organized within itself. So we've looked at cross sections where we don't see the striations. Now we're going to look at longitudinally sectioned skeletal muscle so that we can actually see the cross striations where it's either darker or lighter based on what's present in those bands. So we have our A bands, I bands, and Z discs to start off with. The A bands, we call them anisotropic, which means that it's going to be birefringent in polarized light microscopy. So when we use polarized light microscopy, these will light up. Now, whenever we look at them under just plain light microscopy, these are going to be very dark bands. So these are our dark bands of the striations. And they come from, of course, A being anisotropic. The I bands, meaning the isotropic for the eye, these are not birefringent in polarized light microscopy. So these are going to be the lighter bands that we see under regular light microscopy. And then finally we have our Z disc. This is going to be a single dark line that goes down the middle of each eye band. So bisecting means that we're going to go through it. So here is our image of a cross section of a muscle fiber. The darker bands are going to be our A band. So here's our A band. Our I band is going to be the lighter colored. That's going to be right about here. And then here's our nuclei, of course. Now, whenever we get a little bit closer to it, rather, that was about 200x, this is probably about 400x, we can actually see a little bit more of our A and I band. So here's our A band, and then here's our I band. Now the dark line that's going to go all the way down it, that is our Z disc, and we'll get to see that a little bit more in the next. So here's our electron microscopy picture of it. It allows you to better see the whole picture and actually have each individual part more visible so that we can label it. So again, here's our dark line that's going to bisect our I band. So this is our Z disc all the way down. Here's our A band. One thing interesting about the A band, and we're going to get into this in a little bit later, is there's a portion that's a little bit lighter colored than the rest of the A band. This is called the H. And then we have our Z disc again, I, and it's just going to continuously alternate like this. So whenever we're looking at an image from each Z disc, that is one unit, and we call these sarcomeres. So from one Z disc to the next is a sarcomere. Now the myofibrils are going to be long bundles of these cylindrical filaments, and they're going to run parallel to the long axis. So what that means is they're going to be going like this is our striations, and our myofibrils are going to be going like this because they're going to be parallel to the longer axis. So our muscle is going to be more like this shape, and so the longer portion is going to be from this to this direction. So all of our myofibrils are going to be running parallel to that. And this is just going to be consisting of sarcomeres repeating over and over and over again. So here is our muscle fiber. The sarcomeres are going to be repeating again all the way through the muscle fiber, parallel to the longer axis. And we're going to have our Z-disc present, our Z-disc there, and this is going to be one sarcomere. And our H zone within the A band with the M line, and we're going to get into all those in a little bit. So when we're looking at this, what is causing all these colors or darkness versus lightness to occur? Well, this is due to the overlapping of our myofilaments and our actin filaments. So the I band, which is our lighter band, is going to only contain our actin and our titin, which we'll get into in a little bit. The A band is going to contain both actin and myosin, so it's going to have more protein present, so this is going to cause it to have that darker color. The H zone, which is a little bit lighter, only contains myosin, and our Z disc is just a protein backbone for our sarcomere. So again, due to the arrangement of the myosin and actin band or filaments, this is what causes it to be lighter versus darker under microscopy. Now, the thick myosin, which is going to cause the A band to be darker, is composed of two identical heavy chains and two pairs of light chains. So there's quite a bit of protein found here. The heavy chains are very thin and rod-like proteins that are going to twist together. And as they twist, that is what's going to cause contraction. The light chains are going to be on here and form a head at each end of a heavy chain, and this is going to bind your actin and ATP, which is going to cause the contraction to occur. Now, the actin filaments run between our myosin filaments, and these are G-actin monomers, and they're going to contain a binding site for myosin. So actin filaments are going to be binding myosin and contain a G-actin monomer. The actin filaments are going to be anchored to our Z-disc by alpha-actin, actinin, which is just an actin-binding protein. So if you remember from the image, we had our Z-disc, and this is going to bind our actin molecule. And there's two proteins that are important for our actin molecules. Tropomyosin, which is located in the groove between the two twisted actin strands, and troponin, which has 3 subunits, and you need to know these 3 subunits. TNT attaches to tropomyosin, which is up here. TNC binds to calcium, and TNI is important for regulating the actin-myosin interaction. So if you remember the first letter of each word, you have your 3 subunits of troponin. So, I bands are going to consist of only the portion of thin filaments that do not overlap with the thick filament. Like I showed earlier, this is what causes it to stain lighter. Now, within the I band, there's another protein, which is the largest protein in the body, and that's going to be titin. So if you see this on the test, it's pretty easy to remember, well, titin is another word for very large, so titin is the largest protein of the body. And this is going to help support those thick myofilaments and connect them as well to the Z disc. The A band is going to consist of both thick and thin filaments overlapping each other. Now, within the A band, there's a lighter zone, which I kind of pointed out earlier, called the H zone. This is where there's no overlap, but it's still darker because it contains myosin filaments, so those are going to cause it to be a little bit darker than the I band. Within the H zone, there's an M line. This is going to contain the myosin binding protein that's going to hold those filaments in place. So this is what's going to give that support to the myosin fibers. Whereas the actin fibers have the Z disc, the myosin fibers have the M line. Now, another important part of the M line is creatinine kinase. This is what's going to supply your muscle with the ATP it needs to undergo contraction. So here's that image again. Now we can better look at it and be able to determine every little piece here. So we have our sarcomere. Remember, that's going to be from one Z disc to the next. Our M line is going to be supporting our myosin fibers, filaments. Our H zone is where there is no overlap within the A band, which is the darker band. The darker part of the A band is where there is overlap. Then we have our I band, which is the lighter color. This is going to be where our actin and titin is located. And then these are going to be bound to our Z disc for support. Now, this is our artist rendition of it. Here's what it looks like under the under electron microscope. This is the picture you need to know for the exam. All right, so now that we've gone through how the muscle is organized, now we're going to go into the sarcoplasmic reticulum and the transverse tubule system. Now, remember, the sarcoplasmic reticulum in muscle cells is the same as the smooth endoplasmic reticulum in normal cells. And these are specialized for sequestration of calcium ions. So it's going to store all this calcium until it's needed. When it's needed is whenever it becomes depolarized and releases this calcium of the sarcoplasmic reticulum due to motor nerve synapses on the sarcolemma. So we're still looking at synapses, just like last chapter, causing depolarization. But instead of it being sodium and potassium, we're going to be looking at calcium ions being moved. So the sarcolemma, which is the same as the plasma membrane in other cells, is going to be folded into a system of transverse or T tubules. Now, this is going to allow for uniform contraction and simultaneous release of calcium. So when these become depolarized, since they're all nice and folded, it's going to cause an immediate release all at once of all the calcium. In these guys, you'll find them encircling the myofibrils near the A and I band boundaries primarily. This is because the A and I band boundary is where the actin myosin is going to be interacting with each other, and calcium through TNC, troponin TNC, is going to bind this to allow for contraction. So here's an electron microscope image of it. Here's your tubules going down, folding in, and then here's your muscle fiber, or your muscle fibrils. There we go. So here's our Z disc again, I band, A band, and then here's our troponin and our tubules and TNC, all located right around there. All right, so now that we've gone through all the players in contraction, how does contraction actually occur and what is the mechanism behind it? During contraction, those filaments do not change their lengths at all. So it's very important that you know they do not change. Rather, they're going to increase the overlap that you see. So we're going to see filaments sliding past one another to be closer together. This will bring our Z disc and our M line closer together in the muscle fibril. So for induction, this is going to come from, of course, a neuron, a neuromuscular junction, is going to cause an action potential at the synapse, and this is going to cause the release of acetylcholine. Remember, last chapter I told y'all that's one of the neurotransmitters you need to know, and this is why, because you're going to see it quite a few times here. So acetylcholine is going to be released into the neuromuscular junction, and that's going to cause the action potential to be transmitted from the neuron to the T tubules of the sarcolemma and eventually to the sarcoplasmic reticulum to cause calcium release in the area. Alright, so now contraction is going to occur because calcium ions are going to bind troponin. Remember, T and C is what binds our calcium for troponin. This is going to change the shape of troponin. Now, tropomyosin, which is bound by T and T to troponin, is then going to be moved due to the troponin change in shape and expose active sites. This will allow our myosin heads to attach to those exposed active sites of actin. Now, the myosin heads cannot bind the actin while the troponin-tropomyosin complex is there. So the only way this to occur is through the release of calcium ions. Once this occurs, myosin is going to undergo a conformational change, which just means it's going to change shape, and it's going to pull those thin actin filaments further into the A band, so pull it closer towards the Z disc. And this is going to all require ATP to occur. Now, once that occurs, and once we want contraction to end, or when the impulse stops, calcium ions are going to be removed and brought back into the sarcoplasmic reticulum. Tropomyosin, once T and C loses its calcium, is going to then change its shape again so that tropomyosin, due to T and T binding, is going to recover the active sites. And then the filaments are going to just slowly return back to their relaxed state. Now, rigor mortis, if you remember, or if you've ever heard of it, is whenever someone dies, their muscles tense and are stuck in a contraction-like state. This is due to ATP not being present due to the mitochondrial shutdown of death, and so whenever these form those bridges, it's not going to be able to break apart as easily, so that's why you get that stiff muscle contraction that won't relax. So this image kind of lays it all out for you in steps, and I'm going to go through it with y'all now. The nerve impulse is going to come down the neuron to the neuromuscular junction. That's going to cause release of acetylcholine, which is going to bind to the receptor. This is going to cause depolarization down the sarcolemma and into the T tubules, where it will then affect the sarcoplasmic reticulum. Once it gets there, it's then going to cause the release of calcium ions. Calcium ions are then going to bind to troponin and cause tropomyosin to move, opening the active site, which are these little holes here, and allow myosin to bind to those little heads. Once like that, our calcium's bound and everything, we then pull our Z disc to Z disc, or our sarcomere, closer together so it's thinner than it was at the relaxed state. And then once it's done, calcium's resequestered back into the sarcolemma, and it's going to go back to a relaxed state. So, just to kind of show you the difference, here it is at relaxed sarcomere. Relax. Under contraction, we almost completely lose our I band because of the overlap. So it almost becomes fully A band because there's no opening here. So, innervation and how this occurs with the muscle joints, or not muscle joints, the neuromuscular junctions, is the motor end plate, or MEP, or neuromuscular junction. Myelinated motor nerves are going to branch out within the paramecium. Remember, that's the outermost, or the middlemost layer. The nerves will form synapses with individual muscle fibers, and then we'll have Schwann cells that are going to enclose this and form our motor end plate or our neuromuscular junction. This is the word I want y'all to know primarily. Acetylcholine vesicles will be in the nerve, and this is what they're going to release to cause the signal to be sent on to the muscle fiber. The sarcolemma in that synaptic cleft, so remember, a synaptic cleft is where we have our neuron and our muscle fiber. This is our synaptic cleft in between the two, and there's going to be some folds that are going to increase the surface area of our muscle fiber. So this will allow for more acetylcholine receptors, so the acetylcholine will activate as many as possible. So here's our neuromuscular junction or motor end plate, our nerve fiber going down, and our Schwann cell, and then our skeletal muscle. Here it is with a nice little artist rendition and a electron microscope image. We have our Schwann cells that are going to cover it and cause a control of the environment. We have our junctional folds, and we have our acetylcholine vesicles. These are going to be released, activate, and that's what's going to cause the signal transduction. So, the nerve action potential is going to reach the motor end plate or neuromuscular junction to cause the release of acetylcholine into the synaptic cleft again. That's going to diffuse across the cleft and bind to acetylcholine receptors on the sarcolemma, and this is going to cause an influx of sodium ions, which is what causes the depolarization. The sarcolemma depolarizes, and this is going to cause the production of our muscle action potential. The acetylcholine is going to dissociate from the receptor pretty quickly and either be broken down by acetylcholinesterases, which are enzymes. Remember, ACE is enzyme, acetylcholine, and it's going to be breaking apart the ester of the acetylcholine, or it's going to be retaken up back into the presynaptic neuron. So one single axon can form a single MEP or neuromuscular junction with a single muscle fiber to provide very precise control of muscle activity, and these are going to be in your eyes primarily. Or it can branch to form muscular or neuromuscular junctions or motor end plates with many fibers, 100 or more, and this is going to cause larger muscles for coarser movements, your legs and those things. This is what we call a motor unit, is whenever one axon branches to form MEPs with many fibers. So, muscle spindles are found within our muscle fascicles. And these are special types of cells or structures that are going to be for detection. Now, primarily, these guys are going to be detecting stretch and prevent from a muscle from stretching too far. Now, these are going to be encapsulated with intrafusal muscle fibers, which are just thin muscle fibers with nuclei and interstitial fluid. So, whenever we change our length, which is distension, of the extrafusal muscle fibers, this is what it's detecting. So if it gets stretched too far, it's going to detect it within the muscle spindle. So here's our muscle and better image, so it kind of shows you the nice tapering at the end. But we have our nerve afferent. So remember, afferent goes to which? Goes back to the CNS or goes out to the muscle? And that's going to be, it goes back to the CNS. Afferent receives signals. Efferent is going to send signals. So, afferent, so it's going to be detecting stretch within here. We have our intrafusal muscle fibers here, our stretch receptors, and then our extrafusal muscle fibers, which are our normal fibers. This is our muscle spindle, and that's going to be detecting stretch on our muscles. So here's our little electron microscope image of it with our muscle spindle. And our tendon organ, the muscle spindles, are going to have afferent sensory and efferent motor nerves associated with our intrafusal fibers. So here we're going to have our axons, our myelinated axons of the capsule. And so here's our nice capsule that's going to surround the whole muscle spindle. And then here's our intrafusal muscle fibers. And then our muscle satellite cells again. And this is kind of what it looks like when you do a cross section, look at it under the electron microscope. So, in addition to our muscle spindle, we have a Golgi tendon organ. Very similar role, but it's going to be more smaller encapsulated structure, and it's going to enclose sensory axons again. And this is going to detect changes in tension within tendons primarily. And this is going to be due to muscle contraction. So that if a tendon becomes too excessively stretched, it's going to inhibit the motor nerve activity. So, now that we've gone through that, we're going to go through the types of muscle fibers that we'll see within skeletal muscle. There are 3 different types, and they're going to be named type 1, type 2A, and type 2B. They're going to be based on 2 key characterizations: the speed and the length of contraction. The speed will be from slow to fast, and the length will be from glycolic to oxidative. So the slow oxidative type 1, so we have slow oxidative, is going to be rich in myoglobin, which is going to cause it to be very red. This is due to it containing iron and storing oxygen. All right. Your ATP for slow oxidative type 1 is from oxidative phosphorylation, which, if you remember from your other courses, is how ATP is produced in all your cells. The myosin ATPase activity is low, so it's not going to cause a whole bunch of long contractions and very fast. And these are going to be in your back muscles primarily for stabilization. Now, type 2A is fast, and it's going to be a combination of oxidative and glycolic. It's going to be, again, high myoglobin content. The ATP is mainly coming from oxidative phosphorylation, a little bit from glycolysis, and the myosin ATPase activity is going to be high, so it's going to be much quicker. And these are going to be in your leg muscles primarily. And the last one is type 2B, fast glycolic, and it's going to have a low myoglobin content, so it's going to be more white than red. And ATP is going to be through anaerobic glycolysis rather than oxidative phosphorylation, because we don't have that oxygen coming from myoglobin that we have in the other types. The myosin ATPase activity is going to be high. That's where our fast is coming from. And again, these are for your eyes primarily. So, when we look at a cross section of muscle fibers and we're looking at primarily the types of fibers that are there, whether they're type I or 1, type 2A, or type 2B, we can see that our slow fibers are going to be much darker than our fibers that are type 2A. They're going to have myoglobin, but not as much as our slow fibers. And then the faster ones are going to be very light colored because they're almost going to be white because they lack that myoglobin content that the other 2 types have. So this is table 10.2 in your textbook on page 204. This is going to kind of break down those 3 types of fibers for you. And this way you can go back and kind of review it so that you know where they're from. Now, of course, glycolytic fibers don't have much mitochondria or capillaries because they're not doing oxidative phosphorylation. It's mainly glycolysis, which is done in the sarcoplasm, not in the mitochondria. So kind of look at this table and kind of help figure out why these fibers are acting the way they do. Alright, so we're done with skeletal muscle, which means we're almost done with the lecture because the other parts either overlap or there's not much new to say. So cardiac muscle is very similar to our skeletal muscle in that they contain cross striations. However, in developing, they don't fuse into multinucleated cells. Rather, they form complex junctions from one cell to the next. And they're only going to have one nucleus that's going to be centrally located. Now, these cells are going to join together irregularly, and they're going to have this nice little intercalated disc, which is going to stain very dark whenever we look at them under the light microscopy. Now, the intercalated discs are transversely binds between two cells, and they're going to be desmosomes primarily with some fascia adherents. These are similar to zonula adherents or adherent zones, but rather they're going to be, instead of forming a ring, it's just going to be a spot. This is going to prevent our cardiac muscle from pulling apart during any sort of contraction, because your heart is constantly beating and you don't want that contraction to cause your muscle cells to rip apart. Now, whenever we look at them longitudinally, there's going to be a lot of gap junctions. This is going to allow cell-to-cell communication, which will allow that rhythmic contraction that we see in our cardiac muscle and allow for a nice ionic continuity between cells. So here's our cross section of a cardiac cell with our intercalated disc nicely zoomed in, where we have our desmosomes located, and this is going to help hold the two together. And notice that there's going to be little folds of our intercalated disc to increase surface area to allow for more cell-to-cell contact. And then here's our gap junctions, which are going to allow for that ion consistency throughout our cardiac muscle. And of course, our nucleus is going to be centrally located within the cell. So when we look at it under the microscope, we have our nucleus and our intercalated disc, and notice that it stains very dark compared to the rest of the image. And you can still see your cross sections a little bit in that image. And here is the electron microscopic image of it, where it kind of shows your mitochondria here with our sarcoplasmic reticulum. And then here's our desmosomes and our intercalated disc. So that's it for cardiac muscle. Pretty easy for this. We'll go more in depth on this in the future when we go, actually in the next chapter when we get into this circulatory system. So smooth muscle is our last type of muscle, and it's specialized for slow, steady contraction. And it's going to be involuntary, just like our cardiac muscle. Our smooth muscle is also known as visceral muscle. So know that term as well as our smooth muscle term. The fibers are going to be elongated, tapered, whereas before they weren't in skeletal muscle, and they're going to be non-striated. And each cell is enclosed by a thin basal lamina and a fine network of reticular fibers. So each of these cells is going to have a single nucleus. It's going to be kind of elongated, and it's going to be present in the center of the cell's broadest part. The cells are going to be linked together by gap junctions to allow for that ion exchange, and contraction's a little bit different in smooth muscle as it was in skeletal muscle. The tissue is not striated, so we don't have those nice little bands that we saw in skeletal muscle. Rather, the thin and thick myofilaments are going to crisscross rather than being overlapping, and this is going to cause fewer cross bridges. And instead of troponin, we have calmodulin. So you need to know calmodulin is found only in muscle, or smooth muscle. And this is going to have calcium-sensitive myosin light chain kinase, which is going to be important for our ATP. So here we have our image of our smooth muscle, with the IC being the inner circular area and our OC being the outer layer. And this, because they're crisscrossing, they're going to be cut differently. So our IC is cut lengthwise, and our OL is cut longitudinally or transversely. And you'll only see a few of nuclei, but notice that they're nice and elongated in the center of the cell. And on the right side, we have our cross section, which is going to be denoted by XS. And then we have our longitudinal section there. And of course, we have our perimysium located around it and forming those nice little fascicles. And then here's our muscle, smooth muscle, stained with for reticular fibers, so that we see all the nice little how the reticular fibers are organized to hold the muscle together. And here's our electron microscopy image of it to show that there's not very many mitochondria. So you have about 4 or 5 located on here, and that the unmyelinated nerve is going to come in and it's going to be between the cells, and that's going to be what innervates. And then lastly, here's a longitudinal showing dense bodies, which are thin filaments and thick filament locations. This is our contractile part within our smooth muscle. So our dense bodies are going to be what are Z discs in striated muscle. This is going to be alpha actinin, so it's going to bind our actin filaments. Our thin and intermediate filaments are going to attach to our dense bodies, and it's going to help transmit the contractile force to adjacent cells. So the way actual contraction works is it's involuntary again, and we're not going to have those nice motor end plates that we saw in skeletal muscle, but rather the control is going to be done by autonomic nerves, so it's involuntary, our hormones and other substances, and just local physiological conditions. So certain degrees of stretch will cause muscles to constrict for smooth muscle. Nerve supply is primarily just to modify it, not initiate it. So remember, nerves in smooth muscle is for modification of the activity, does not initiate it primarily. So here's our artist rendition. We have our smooth muscle cells. Notice that the nucleus is in the center of the broadest part. And here's our dense body with our alpha actinin binding our actin molecules, and then our myosin being in between, binding 2 dense bodies together, so that when contraction occurs, it makes the cells look fatter rather than relaxed. Alright, so the last section is how our muscle cells regenerate. Skeletal muscle does not undergo mitosis. It's very important that you know that this does not occur. So there's very limited regeneration of skeletal muscle. Rather, it comes from the few satellite cells that you'll find within your muscle tissue. These are inactive cells that will become activated only after injury to proliferate into new fibers. Cardiac muscle does not have the satellite cells, so there's even less regeneration that can occur past your childhood. So any defects or damage to the heart is only going to be replaced by fibroblast forming scars. This is why whenever someone has a heart attack, it's very important to get them treated as quickly as possible, because you're not going to get new heart muscle to form. Rather, you're going to get scar tissue to form. And then lastly, you have your smooth muscle. This is probably the most capable of undergoing regeneration. So they have a much more active regeneration response. After injury, viable muscle cells undergo mitosis to replace damaged tissue. So smooth muscle is really the only one that has any sort of regeneration capabilities. Skeletal muscle has those satellite cells that are kind of scattered around to kind of help a little bit, and cardiac muscle has nothing. So you need to remember those 3 different types of regeneration that can occur with your muscle tissue. So on page, let me find it real quick, table 10.1 on page 202, I recommend reviewing this quite a bit for your final. or not for your finals, but for your next test, because this is going to break down the 3 different types of muscle tissue and compares them. So you have your fibers, your striations being absent in smooth muscle. The nuclei are going to be central, peripheral, are controlled contraction, troponin C, similar to skeletal muscle, and this is going to be myosin phosphorylation by MLCK. And then the second part of the table tells you how the connective tissue is organized, where you can find it, the function of it, how it's innervated. So remember, motor neuron end plates, autonomic. So these are involuntary, how the contractions occur, cell response to load, and capacity for regeneration. So that's it for chapter 10. I will be posting a review on Blackboard shortly and a practice quiz over just what we just covered.